When a problem arises, people turn to scientists for answers. The University of Washington 2006 Science Forum features faculty sharing research results on the world's most vexing problems. Mary Desis examines how the immune system may be used in the future to prevent cancer through the development of vaccines. David Eaton looks at the research on genetic, lifestyle, dietary, and environmental factors to puzzle out why some of us get cancer and others don't. Garrett Odell examines the use of mathematical models to shed light on how biological gene networks operate and how these models reveal the networks to be astonishingly robust, despite their unintelligent design. Buddy Ratner updates us on the progress of regenerative medicine and addresses the practical and ethical issues surrounding tissue regrowth and organ replacement. And Ray Hilborn provides facts to dispel the fears of a worldwide fisheries crisis and discusses the changing objectives of fisheries management. Join us in Puzzling Out the Science. Exploring Our World, Puzzling Out the Science is made possible by the University of Washington Alumni Association, the College of Arts and Sciences, the Office of Research, the College of Engineering, the School of Medicine, the College of Ocean and Fisheries Sciences, and the School of Public Health and Community Medicine. Additional funding for this program is provided by the University Bookstore and UW Medicine.
Welcome to the first lecture of the 2006 UW Science Forum. My name is John Slattery. I'm the Vice Dean for Research and Graduate Education in the School of Medicine. And I'm thrilled to be here today with you. UW Science Forum was created to provide information and insight to the community from top UW researchers on new developments in science and technology. I'm pleased to see in the audience this evening donors, UW alumni, friends of the College of Arts and Sciences, the Office of Research, the School of Medicine, the College of Engineering, the College of Ocean and Fishery Sciences, and the School of Public Health. I'd like to tell you a little bit just about UW Medicine. It's a name that you've seen around, but I hope that you know that that UW Medicine actually owns or operates several entities, including Harborview Medical Center, the University of Washington Medical Center, which I'm sure you'll know has been rated recently as one of the top 10 in the US, the University of Washington School of Medicine, which happily actually serves 5 states, not just the states of Wyoming, or the states of Washington. It serves Wyoming, Alaska, Montana, and Idaho as well. The school, as you know, I think, has been rated in the U.S. as tops, actually, in primary care and rural medicine. And that spectrum of activity actually extends, of course, to being absolutely top flight internationally in biomedical research. We house 5 Nobel laureates, 25 members of the Institute of Medicine, 26 or so when Nora gets there, and 25 members or 26 members of the National Academy of Sciences. We also operate UW Medicine clinics in the neighborhoods around the Seattle region and UW Physicians. UW Medicine shares in the governance of Children's University Medical Group and the Seattle Cancer Care Alliance. You can see why this is just a great place to work. And what I'd like to do now is introduce Dr. Nora Desis. She received her MD degree from the University of Nebraska Medical School and completed a residency and chief residency in internal medicine at the University of Illinois in Chicago. She held a fellowship in oncology at the UW and the Hutchinson Cancer Research Center, and she is an expert and pioneering investigator in breast and ovarian cancer immunology and translational research. Nora is a member of Alpha Omega Alpha and the American Society of Clinical Investigation. She's a member of the AACR Immunology Task Force, a deputy editor for translational oncology for the Journal of Clinical Oncology, and chair of the National Cancer Institute's Clinical Oncology Study Section. She also is the director of the UW General Clinical Research Center and the director of the Center of Translational Medicine and Women's Health here at the university. Her multifaceted research program with the Tumor Vaccine Group includes the discovery of new antigens for breast and ovarian cancer and the development of vaccine and cellular therapy targeting of those antigens. Tonight she'll be discussing that research. Let's welcome Nora.
Thank you. Well, this evening I'd like to talk to you a little bit about vaccines, and I'm putting them in the context of targeting breast and ovarian cancer. But the principles that I'm going to talk about tonight really can be applied to any different type of immunotherapy against cancer. I'd like to first start by talking a little bit about immunity in cancer. Does the immune system even play a role in tumor eradication? And then talk a little bit about vaccine design. So we're used to going to the doctor and getting vaccines like tetanus shots and flu shots. What's different about a cancer vaccine? And then finally, I'd like to end up just talking a little bit about the clinical applications of cancer vaccines, giving you an example from our group targeting an antigen that's expressed on breast cancer called HER2/neu. So when you talk about generating an immune response, especially tumor-specific immunity, you really have to think about the generation of an immune response in a continuum. First, you have the tumor, and that's represented here. And the tumor is expressing proteins. And if these proteins are immunogenic, meaning that they can stimulate an immune response, they're termed antigens. So you have a tumor sitting there, and you have some immunogenic proteins being expressed by the tumor. What you need to have now is antigen-presenting cells, basically being able to pick up the portions of the tumor, actually living cells that will go and chew up bits of the tumor and present it to immune system cells in the draining lymph node. So much like you'd have a cut on your skin, antigen-presenting cells will come in, clear up the bacteria in that cut, and then traffic to the lymph nodes that are near that cut and present those antigens to T cells in the draining lymph node. And T cells are really described as two different types, cytotoxic T cells and T helper cells. Cytotoxic T cells or lymphocytes are the immune system cells in your body that actually can really respond to viruses and basically latch on to a cell that's infected by something, be it a virus or oncogenic protein, let's say, and actually result in killing the cell. But the other type of lymphocytes that we have are called T helper lymphocytes. And those lymphocytes are described as either Th1 or Th2 lymphocytes. And they're described as T helper lymphocytes because they secrete natural substances, cytokines, that allow the elaboration of different types of functions of other immune system cells. So if you're a Th1 lymphocyte, you create a very inflammatory environment. And that's really what we'd like to get in the tumor bed of cancer, making cancer look very dangerous. If you're a Th2 lymphocyte, you dampen the immune response. So in cancer, we're really looking to see CD4 T cells that are Th1 inflammatory T cells that have been stimulated by the appropriate antigen-presenting cells. Once you have T cells in the draining lymph nodes stimulated by antigen-presenting cells, they will go then and travel back to the tumor and start killing the tumor cells. And this is really the basis of the generation of any type of immune response. You have to have an immunogenic protein picked up by antigen-presenting cells, which stimulate T cells, to go back to wherever the antigen is, be it a virus or be it a tumor, and then our immune lymphocytes really start trying to kill those tumors. So that is how, theoretically, we would develop tumor-specific immunity. What real data do we have that the immune system has anything to do with cancer? Well, we know that if you're immunosuppressed, that increases your cancer risk. So in studies of people who've got kidney transplants or liver transplants or lung transplants, we know when they take agents that really dampen their immune response, they're more prone, very much so, to developing different types of cancer. Not only cancers like lymphoma or skin cancer, but also run-of-the-mill cancers like colon cancer and breast cancer. So this is inferential evidence that if you take the immune system away, somehow cancer begins to start. We also know that spontaneous regressions of cancer occur. And many of you in this audience might know people who said, oh yeah, I got diagnosed with cancer, and then I went back to my doctor maybe 2 weeks later and the cancer was gone. That isn't a story that really truly does happen, and in fact it's been reported with multiple different types of cancers. This is just a number of reports in the literature: colon cancers, lung cancers, breast cancers. In fact, documented spontaneous regressions of cancer occur so frequently that you can't really report them in the literature anymore. They're not novel. So obviously people think that if cancer regresses spontaneously, there must be some natural mechanism at play, and perhaps that mechanism is the immune system. We also know that tumors can actually have infiltrating lymphocytes attacking them. So in many cases, if you go to the doctor and you have cancer and you go to surgery and you have that cancer removed, the pathologist can actually see that there's an inflammatory response in the cancer, that your immune lymphocytes that we talked about on the last slide really are trying to attack the cancer. But finally, I think the biggest evidence that's come that the immune system really has something to do with cancer is in the last decade, we now know that human tumors are immunogenic. So we know that patients with cancer can mount an immune response against their cancer. And in fact, we know many of the proteins that are expressed on cancer that are involved in stimulating that immune response. So I'd like to show you an example in ovarian cancer of exactly what I'm talking about. This is a paper that was published a few years back by a group at University of Pennsylvania. And the investigator asked a very simple question. He had been following ovarian cancers for a long period of time, and he said, well, we know that T cells do infiltrate the tumors, and I've got this big cadre of patients in their tumors that I've been following for a long time. Does that T cell infiltration mean anything in terms of their overall survival? And what he found was that those tumor-infiltrating lymphocytes, which you can see with these very dark dots here, and this is a picture of someone's tumor, really could be described in two different patterns. This first pattern is intratumoral, and what you see here is many of these T lymphocytes are actually burrowing right into the tumor bed. But what these investigators found was that there was another pattern of tumor-infiltrating lymphocytes, and these were called stromal. And what that means is these T cells were not burrowing into the tumor. They seemed to stop at the capsule that surrounded the tumor, and they weren't really being allowed into the tumor bed. So it was very striking. People either had no T cells, or they had intratumoral T cells or stromal T cells. So these investigators went back and they categorized all the ovarian cancer patients. There were actually 174 of them. And most of the patients had advanced stage disease, as many ovarian cancer patients are diagnosed with more advanced stage disease. And when they simply separated patients based on whether they had intratumoral T cells or no intratumoral T cells, the difference was striking in terms of the overall survival, with those patients that had evidence of the T cell burrowing into the tumor bed having a much greater prolonged survival than those patients who didn't have evidence of that immune response. Furthermore, if they took the one thing that really makes an impact on overall survival in ovarian cancer, which is at the time of the initial surgery, having what's called optimal debulking, that means all the cancer has been removed to the best of the ability of the surgeon, to the point where they can't see cancer anymore in the abdomen. Having intratumoral T cells actually was a better predictor of overall survival than having suboptimal debulking. So in those patients who didn't have any intratumoral T cells, but yet had all the tumor removed at the time of surgery, they statistically significantly did not do better than those patients who did have evidence of the T cells infiltrating the tumor, even when the surgeon was not able to get out every last bit of tumor. So based on this study, people at the tumor vaccine group, the group that I worked in, said, All right, so tumor-infiltrating lymphocytes are kind of nonspecific. It just means that your immune system is trying to have an immune response against your tumor. What if your immune system was actually responding to some of these immunogenic proteins expressed by the tumor? And so what Vivian Goodall in the group did was she collaborated with folks at the Hutch who had created a big bank of sera, blood, from patients who were newly diagnosed with ovarian cancer. In fact, many of these patients didn't even know if they had ovarian cancer at the time this blood was drawn. They were going for initial surgery for an abdominal mass. And what Vivian found was, if you looked at, let's say, immune responses to p53, which is a cancer-causing protein, those patients who had immunity to p53 at the time of diagnosis, even of advanced stage ovarian cancer, had a better overall survival than those patients who had no evidence of immunity. Here's an example of immune responses to HER2/neu. And one of the things people often say is, well, perhaps being able to mount an immune response predicts that people are just more healthy and they're going to do better with their cancer anyway. But if you look at immune responses to something like tetanus, which is an immune response everyone has here if they've had a tetanus shot, there was no difference or survival benefit in those people who had tetanus-specific immunity. So this is the type of data that's being collected now in the field of tumor immunology that suggests that, yes, there is an active immune response against cancer, and that immune response may have an impact on overall survival. So how does tumor-specific immunity develop? Well, you start off with a cancer cell, and as we talked about before, you have antigen-presenting cells that are either picking up necrotic proteins that are being shed from this cancer cell, or actually these antigen-presenting cells, such as dendritic cells, and we'll talk about these a little bit later, will phagocytize or eat the cancer cell. Because around every cancer cell or in every tumor bed, there's a certain amount of low-grade inflammation that's signaling the immune system to start doing a cleanup. When these dendritic cells or antigen-presenting cells get activated, they go to the lymph nodes. They, again, present the antigens to the T lymphocytes, either cytotoxic T cells or those T helper cells, and then these antigen-specific T cells will go back to the tumor bed and start proliferating and growing and trying to kill the cancer. Now, one of the interesting things is that many of the immunogenic proteins that are expressed in cancer are not abnormal proteins, they're self-proteins. And we've now come to realize that one of the reasons why people with cancer have immune responses against their tumors, but also have cancer, is that many mechanisms of immune escape are really causing those immune responses to be non-functional. So let's go back to that circle that describes the generation of an immune response. We know that tumor antigens are present on tumors, that proteins that are immunogenic are expressed on the surface of tumors or inside tumors. And we also know that many of these proteins are self, they're part of our self. They're not viruses, they're not foreign, they don't give a foreign stimulus to the immune system. So tolerance is one of the major mechanisms by which cancer escapes immune surveillance. Our own natural mechanisms of trying to prevent us from developing autoimmunity prevent us from making a rip-roaring response against cancer. So cancer, these proteins that are immunogenic, are aberrant enough that our immune system recognizes that they're abnormal, but they're not foreign enough that a very destructive inflammatory response is made against them. When we talk about the antigen-presenting cells that really attack cancer and chew up the cancer, now that we've studied many tumors, we know that many of these antigen-presenting cells are really defective. They don't have the appropriate co-stimulatory molecules on their surface that is needed to really stimulate a T cell response. And in fact, we know the tumors themselves secrete substances that cause the antigen-presenting cells to be defective. Furthermore, we know that the most potent antigen-presenting cells in our bodies are called dendritic cells. In fact, they're so potent in being able to stimulate immunity that they're less than 1% of the circulating blood cells in our body. Most of them are found in our skin, which is really the first avenue of invaders into our body because they're able to stimulate an immune response to almost anything. It's rare to see a dendritic cell in a tumor bed. So we know that the antigen-presenting cells in the tumor are defective, and the very potent ones that we really need to stimulate immune responses to self are not there. When we talk about the T cells being stimulated in the draining lymph node, and we talked about the cytotoxic T cells and then those T helper cells, we know now by studying T cells in cancer patients from the draining lymph node that those very inflammatory Th1 T cells are not there. And in fact, many of the T cell responses against cancer are very low level, and I'll show you that in a second. So even though they can be stimulated to some regard, if you look at a cancer-specific immune response compared to immune response against a foreign protein, like a virus, the difference in magnitude are logs different. And finally, if we look at those T cells trafficking to the tumor, and we look at the tumor microenvironment, as I alluded to before, the tumor secretes substances that actually dampen the immune system. So they secrete cytokines, they generate different types of stromal factors or proteins that are actually secreted by the tumor that prevent the T cells from even working. So when you think about trying to design immune-based therapy to have an effect or stimulate very successful immunity against cancer, not only do you have to think about what type of immune response you want to generate, you have to think about how am I going to modulate the microenvironment so that if I can get T cells homing to the tumor, they won't go into that microenvironment and be turned off by the tumor itself. And I think this is the biggest problem with cancer vaccines over the last multiple decades that these vaccines have been studied in patients, is that most of the time, cancer vaccines have been used as therapeutic vaccines, meaning that they've been tested in patients who have existing cancer that isn't responding to any other treatment, and people will immunize cancer patients, and unfortunately, cancer vaccines have not had much success. And part of that is due to the fact that we know that we can take a vaccine and stimulate an immune response, but when those T cells home to the tumor, because of the suppressive microenvironment, there's really minimal tumor cell death and, in fact, continued tumor cell growth. So with this background, as we move forward into thinking about vaccine design, all these factors have to be taken into account as we try to figure out what types of vaccines would be most effective at circumventing some of these problems with the immune response and stimulating an effective immune response to cancer. So unlike many of the vaccines people have received that really consist of just taking proteins and mixing them up in a substance that make them more immunogenic, cancer vaccines have to be designed to also combat some of these potential problems. What I'd like to do talking about vaccine design is kind of couch it in a story of targeting a particular antigen or immunogenic protein called HER2/neu. HER2/neu is a protein that's overexpressed in breast cancer, and here's a picture of using antibodies to stain the surface of these cells. And here you can see that the HER2/neu gene is amplified in breast cancer cells. And so the HER2/neu protein is expressed on the surface of breast cancer cells. And if you think about HER2/neu overexpression in breast cancer compared to some of the normal cells where HER2/neu is expressed at basal levels, in cancer cells, it's overexpressed maybe 50 to 100-fold. HER2/neu is a normal cellular protein that's used in cell growth and division. And so basically in gut or other rapidly dividing cells like the cells in your skin, it also is overexpressed because it's used to cause those cells to grow and divide. When we look at the level of immunity against HER2/neu, we realized several years ago that the more protein overexpression of HER2/neu patients had, the more likely they were to have an immune response. So if you look at patients who have basal levels of HER2/neu compared to those patients who have minimal levels of HER2/neu expression, compared to those patients who have marked overexpression of HER2/neu, those patients who have marked overexpression of HER2/neu are more likely to have really significant levels of immunity. Now, why would that be so? What is it about overexpression that makes a protein immunogenic? And it really, again, has to get down to how T cells recognize proteins. They don't really recognize proteins as globs that are floating around in your immune system. As I said before, antigen-presenting cells uptake and process these proteins into fragments called peptides. And the way your immune system really makes these fragments is dictated genetically by the type of immune system you have. So for example, if you have a virus, let's say a herpes virus in your lip, and basically you want to generate an immune response to it because you want to be able to limit that herpes virus in your lip, the way that your immune system works is by chopping up those viral proteins into small fragments or peptides and presenting them in immune receptor molecules called MHC molecules that are on the surface of the cell. T cells will dock on those MHC molecules and recognize those peptide fragments, and they'll end up killing a tumor cell. But back in the early 1990s, a group in Germany found that if you looked at resting cells, like skin cells, and MHC molecules that are expressed on those resting skin cells, those resting cells that are non-infected, the MHC molecules or immune receptor molecules are chock full of peptides, yet you're not having an immune response against your skin or other organs that are non-infected. And so these investigators were able to take peptides and sequence them and find that these peptides in resting cells were really normal self-proteins like parts of blood or albumin. And what they found was your immune system will preferentially put dominant epitopes of self-proteins in immune receptor molecules to kind of prevent yourself from attacking itself. And that these dominant epitopes of self-proteins don't elicit an immune response. But other investigators looking at animal models of autoimmunity, where they're able to turn on a protein so it's overexpressed, kind of what I showed you in HER2/neu, discovered that subdominant epitopes of these self-proteins could elicit an immune response. So the reason why that data that I showed you, that patients who had increasing levels of HER2/neu expressed on their surface of the tumor could stimulate an immune response against HER2/neu, was that suddenly the peptide repertoire display in their MHC molecules on the surface of their tumors was different, and that subdominant peptide epitopes had been shielded from the immune system, probably because normal self-proteins aren't overexpressed 100-fold like HER2/neu is. And so that overexpression of a self-protein in cancer is a way to make that self-protein a tumor-specific antigen. So one of the major problems, although patients with HER2/neu overexpression can have an immune response against their tumor, is that the immune response, as I told you before, were extremely low level. So here's an example, again, using a tetanus shot of anyone in this room, and then looking at HER2/neu antibodies in breast cancer on the far slide. As you can see, tetanus immunity in anyone in this room is really quite high, and this is an example of antibody immunity. In fact, it's 100-fold higher than HER2/neu antibodies that you'd find in a breast cancer patient. So many people would say, well, you just told us that cancer is kind of immunosuppressive, so maybe it's because their immune systems aren't completely functional. But actually, we looked at some patients who, during the time they had breast cancer, had tetanus shots, and we found that their immune responses to tetanus were no different than anyone who didn't have cancer who got a tetanus shot. And that is something that really has broken the lid off tumor immunology in the last decade, is our understanding that unless a patient really is in the last throes of cancer, their immune systems are really quite functional. And in fact, it's their healthy immune system trying to prevent them from attacking self-proteins expressed in cancer that's really limiting the immune response. So how could we figure out how to increase that immune response to make it effective or inflammatory? And for this, we turn to modeling immunity in new transgenic mice. New transgenic mice are mice that were genetically bred as embryos to express neu or HER2/neu on a promoter that as the mice developed, they would have HER2/neu expression in their breast. And as the mice got older and had pups, they would end up developing breast cancer very similar to patients who had HER2/neu overexpressing breast cancers. And here you can see normal breast epithelium. In panel B, you're beginning to see invasive cancer. And in panel C, that's really advanced stage cancer in the mouse. So this was really a great model to see exactly how we could intervene and potentially stop the development of cancer, because these mice were genetically destined to develop cancer. And another thing that was really excellent about this model was that the mice, since they were born with NEU, were tolerant to NEU. So this was really a model that was almost identical to the way breast cancer was in humans. So we went back to thinking, if we're going to stimulate an inflammatory environment, because we know cancer is preventing that inflammatory environment from developing, well, we want to focus on those lymphocytes. And we know that in breast cancer, those Th1 inflammatory lymphocytes, those CD4 T helper cells, usually aren't stimulated and aren't there in great numbers. So when we thought about developing a vaccine, knowing that for cancer vaccines, you have to attack some of these problems with the baseline immune response, we basically went back to that concept of those subdominant epitopes and tried to figure out those peptides that would be presented in MHC molecules that could be recognized by the immune system. And to do that, we actually used computer modeling systems. And from that, we created several potential subdominant epitopes that would stimulate a CD4 or a Th1 T cell response. And when we went back to the mice and we vaccinated them, we found that we could generate both a peptide-specific response that actually responded to protein. And so I'm just going to show you briefly. This is a tumor cell that overexpresses NEU. This is a peptide. And basically, when you immunize the animals, they develop an immune response to the fragment as well as to whole tumor cells. And in those blue lines, what it's demonstrating is that the T cells that were stimulated by this vaccine actually strongly secreted those inflammatory cytokines that may start to modulate that very unhealthy tumor microenvironment into a healthy tumor microenvironment. Furthermore, if we took the vaccine that we developed from these fragments and we immunized animals, we could protect the animals from developing tumor compared to animals that just got a control vaccine that didn't include these fragments. But the interesting thing, if you think back to what I talked about before with therapeutic immunization and the fact that cancer vaccines haven't been so successful because people have been immunizing patients who really have advanced cancer, when we started immunizing animals on the same day as they began to develop tumor, we didn't have any effect of this vaccine compared to a control alone. So the vaccine that protected animals before they developed cancer, much like a vaccine would protect you from the development of exposure to a virus, when we used it in a therapeutic setting, was not very effective, just like most cancer vaccines. So we asked the question, is there a disease or a place where we may be able to modulate the microenvironment, where the tumor burden isn't so excessive that we might be able to stimulate T cells if they were this inflammatory that may get a handle on the tumor? And when we thought about being able to have almost no tumor microenvironment and being able to eradicate the tumor so that the immune system overwhelms the tumor, not the tumor overwhelming the immune system, we really decided at this point to focus on breast and ovarian cancer. And the reason for this is, when you think about breast and ovarian cancer, they're very highly treatable diseases. The major clinical problems with breast and ovarian cancer is disease recurrence. Even in the most advanced stages of the disease, you can, in many cases, get a complete clinical response with standard therapies alone. And when you look at the time to relapse, it doesn't take place over months, it can take place over years. So we figured if cancer vaccines are going to work, this would be the clinical setting to be able to take patients who have more advanced stage disease but have been treated to a complete clinical response and tried to turn on their immune systems to be able to go to the small places of micrometastatic disease that are probably present and hopefully be able to eradicate those small deposits of micrometastatic disease. And so in the last part of the talk, I'd like to show you an example, again sticking with HER2/neu, taking what I told you about looking at vaccine development and applying it in patients with breast and ovarian cancer to using that peptide-based vaccine approach or that fragment vaccine approach and seeing what we find out about whether the tumor actually can be modulated by the immune system. So if you think back to that picture, we've talked about the antigens, HER2/neu is one. We've talked about looking at the T cells and stimulating Th1 immunity. Now let's talk about the antigen-presenting cells. I told you that one of the problems with the tumors being able to stimulate immunity is that dendritic cells, or very potent antigen-presenting cells, are rarely in the tumor bed. And I also told you that our skin is really the biggest holder of the most potent antigen-presenting cells, dendritic cells, in our body. So when we decided to develop a vaccine to use in breast and ovarian cancer patients, we knew that if we picked these subdominant peptide epitopes, at least in animal models, that was a good way to circumvent tolerance. But we thought what we also really needed to do was use the most potent antigen-presenting cells in stimulating that response. And so what we did was, and in the middle panel you can see a picture of resting dendritic cells. If we used a substance called GM-CSF, which is actually a protein that causes your immune system to get revved up, and it's used to actually stimulate white blood cell growth in cancer patients after chemotherapy. If we mixed that up in the vaccine and we injected it into the skin, that GM-CSF could turn on dendritic cells. And in that final panel, what you see is a dendritic cell in the skin now expressing high levels of the most important co-stimulatory molecules that are needed to present antigen to T cells in the draining lymph node. And so with that, we use computer modeling systems to help us figure out what subdominant epitopes were for human HER2/neu, developed a peptide-based vaccine to try to stimulate those Th1 peptides using GM-CSF mixed up with the vaccine intradermally, and we immunized almost 40 patients with stage 3 and stage 4 breast cancer. And what we found was that almost every patient developed a peptide-specific response, a response to that fragment or subdominant epitope derived from HER2/neu. And almost 60% of patients developed a response to the intact protein. So if you took a tumor cell that overexpressed HER2/neu and put one of these patients' lymphocytes, T lymphocytes, with the tumor cell, the T lymphocytes would begin to kill the tumor cell. So for us, this was kind of proof of principle from mouse to man that subunit or subdominant vaccines could circumvent tolerance, and people could respond to both the fragments as well as intact protein. And so now our question was, would these T cells be able to home to the tumor and begin to modulate that tumor microenvironment into something more inflammatory? And if you think about that circle that I showed you, ending up with the tumor microenvironment, what we really would like to see is those CD4 T helper cells secreting different cytokines that would actually activate that microenvironment to bring in more T cells. And when we looked at some of the T cell clones that we derived from the patients after vaccination, again, many of these clones were secreting high levels of inflammatory cytokines like interferon gamma and TNF alpha. So it appeared, at least in a Petri dish, that we did generate those type of T cells or Th1 T cells that could create that kind of inflammation. One of the things that we found during the course of the study is that patients began to develop immune responses against fragments of HER2/neu that they hadn't been immunized against. And this phenomenon is called epitope spreading. And it means that their immune responses were evolving along with immunization. So here in the panel that's closest to me, you see gray bars that are the immunizing peptides, and white bars are the fragments of protein that people develop T cell immunity to that weren't in their vaccine. We also found out that people during the course of immunization began to develop immunity to immunogenic proteins expressed on their tumor other than HER2/neu. So for example, at the beginning I told you about immunity to p53 in ovarian cancer patients. This is an example on the far slide of 2 patients who were immunized on one of our HER2/neu vaccine trials who had no immune response to HER2/neu or p53 prior to immunization, and at the end of immunization with a HER2/neu vaccine developed immunity to HER2/neu as well as to this antigen expressed on their tumors, p53. Again, this is called intermolecular epitope spreading. So what caused this phenomenon that patients were immunized and had a broadening of their immune response, not only to other portions of the HER2/neu protein, but also to other immunogenic proteins expressed by their tumors? And actually what it means is there was productive immunity modulating the microenvironment, that those Th1 inflammatory T cells initiated an immune response, actually homed to the tumor bed, created the type of inflammation that really activated other antigen-presenting cells in the area, which resulted in tissue destruction and debris. And if you think back to how tumor immunity is started with necrotic cells being able to be picked up by antigen-presenting cells, that is how immunity is spread to other antigens. So what was true in the mouse was now true in women, that you could create that modulation of an inflammatory microenvironment and start having native immunity begin to develop, not only to HER2/neu, but to other immunogenic proteins expressed by the patient's tumors. One of the things that's really the key to active immunization, especially if you're talking about trying to protect patients from cancer relapse for a period of years, is that the immune response would persist. And basically what we found, and this is data generated by Lupe Salazar in our group, is that as long as now, up to 8 years after vaccination, 60% of patients still have evidence of detectable immunity. And here's an example just showing you CD4 Th1 cells that are secreting the inflammatory cytokine interferon gamma. You can see prior to the vaccine, this particular patient who had metastatic breast cancer has really no detectable immunity to HER2/neu. After 1 month after her last vaccine, has about 0.8% of her circulating T cells specific for HER2/neu, but 5 years after vaccination, almost 3% of her circulating T cells are specific for HER2/neu. Now, this was a question for us, whether having that type of immune response meant that her tumor was coming back and she was actively fighting the tumor, or was this a protective immune response that was preventing disease relapse? So what Lupe did is she went back and contacted all the patients on this initial study, now with a median follow-up of 7.5 years. And in our largest cohort, which were stage 4 breast cancer patients, and these are women with metastatic disease, 38% of the patients were still alive 7 years after the end of immunization. So this was good data for us that maybe there was some modulation of the tumor microenvironment going on, and there may be some protective benefit to vaccination. And so with this, we've started a phase 2 study really addressing the question whether immunization against HER2/neu, for example, would prevent cancer relapse. And just recently, one of our collaborators on the East Coast took one of the peptide vaccines and moved it into even an earlier category of patient, those patients with just positive lymph nodes. And what he found, and that's in the panel at the top, is that after active immunization, and he's comparing this group to a control group, that he actually had a statistically significant decrease in recurrence rate or relapse rate in those HER2/neu positive patients who were immunized with a fragment-based vaccine than those patients who got no immunization. So in summary, there are many antigens that are expressed on breast and ovarian cancer. And you can kind of come up with a concept of the development of a multi-antigen vaccine that would be able to potentially protect patients from the development of disease relapse. But unlike infectious disease vaccines that are already seen as very foreign by the immune system, tumor antigen vaccines really need to be formulated so that they can address mechanisms of immune escape. You really want to take the non-productive inflammatory environment that's present in a tumor bed and change it into a productive inflammatory environment, which will allow dendritic cells to differentiate, mature, and increase their functions, being able to present antigens, immunogenic proteins to T cells, and have those T cells be able to be activated and expanded and home to the tumor bed to continue to eradicate micrometastatic disease after optimal therapy. So in summary, I just would like to acknowledge the group, the tumor vaccine group at the University of Washington, of which I'm part. And if you'd like any more information about any of our trials or a lot of the stuff that I talked about today, we actually have information posted on our website. It's right there at the bottom of the page. Thank you. Does anyone have any questions?
Hi. I'm just wondering, what would be standing in the way of allowing women that are at high risk but don't have evidence of cancer to receive the vaccine? What sort of steps are between here and there,
Right.
if you show that you can prevent relapse?
One of the major problems with these proteins, as I said before, is that they're self-proteins. And so basically, when you generate an immune response against HER2/neu, or any of these proteins, p53, you're generating immunity against self. So what the long-term effects of that are, we don't know. And because these immunogenic proteins are unique in that, I think that there has to be a lot more testing of the long-term effects. Very different from, let's say, the cervical cancer vaccine that's targeting a virus. So you know that the immune system is really geared toward HPV and not any other self-proteins that may cause problems later on.
Can you say something about how Herceptin works and whether or not Herceptin could be used in conjunction with this therapeutic vaccines? Could they be used together?
So Herceptin is a monoclonal antibody that targets HER2/neu. And if you think back to the slide I showed you of overexpression of HER2/neu on a tumor cell, the Herceptin monoclonal antibody will latch on to HER2/neu, and it turns off the growth signal of HER2/neu, so it makes tumors more amenable to chemotherapy. We and others have found that Herceptin, which is an antibody, also stimulates an immune response in patients. So monoclonal antibodies have a receptor called an FC receptor that will bring in antigen-presenting cells. And so what we have found is that patients who are taking Herceptin actually augment immune responses to HER2/neu. So right now, we just finished a phase 1 study of giving the vaccine that I talked about at the same time as Herceptin. And the biggest issue that we had was whether there were any additional cardiac toxicities with it, but it was very safe and it was very effective in stimulating immunity. So I think the concept of developing combination therapies that work by multiple different ways but are synergistic is probably going to have a lot of importance in the future of breast cancer treatment.
I think I'm confused. So if HER2/neu is a basal, or if you have a baseline of that anyway, but in cancer, it's overexpressed.
Right.
It's a signal, as it were, of having cancer?
It's a cause, one of the causes of your cell being a cancer cell.
Okay, so that, as an immune response, is the body's way of, is it the body's way of fighting it, or is it just cells that have developed some aberrant behavior?
Right.
And so that's part of my question. The other part is, does being HER2/neu positive have an adverse relationship with traditional chemotherapy?
I'll answer the last question first. Being HER2/neu positive does have an adverse relationship with traditional chemotherapy. And before Herceptin, Having HER2/neu protein overexpression on your breast cancer was a poor prognostic indicator. People did not do well with standard doses of chemotherapy who had HER2/neu overexpressing tumors. Once Herceptin came along, which caused the tumor to be more amenable to chemotherapy, now there's some data that would suggest that patients who have HER2/neu protein overexpression actually do better than other patient categories because of the combination use of Herceptin. Now, what is your immune system trying to do when your tumor cell, or when a cell in your body starts upregulating HER2/neu? The same thing if a cell in your body were to start having lots of p53 in it. Your immune system realizes that this is not normal, and there is an immune response that initially is being generated, but the response is not productive. So it's an aberrant enough of a overexpression is aberrant enough that your body knows that it's wacky and it should be doing something about it, but because these are self-proteins, the signal that the immune response is getting is not dangerous enough that it just starts a full-fledged rip-roaring immune response that will eradicate the tumor. And so many people think that if there's some way early on we can take the reins off that immune response and just make it so it is rip-roaring right away, that might be one way that at the very earliest stages of disease you'd be able to prevent disease from getting out of hand.
Are you finding with the Herceptin and the added benefit of that and other vaccines or whatever, is this going to be protective of other, or protective toward other cancers?
I think the interesting thing about most of these tumor antigens, like let's say HER2/neu, people think about cancers in terms of tissue types. I have lung cancer, I have sarcoma, I have colon cancer. But more and more we're starting to think about cancers as molecular types. So there are sarcomas that overexpress HER2/neu. There are lung cancers that overexpress HER2/neu. So one of the things we're working on in our group is coming up with a multi-antigen vaccine that would work for both breast and ovarian cancer, because many of the proteins that are involved in causing those cancers that are immunogenic as well are the same. So it would be possible potentially to develop vaccines that hit molecular categories of cancer, whether they started in one specific tumor or a tissue or another.
Hi, I was just wondering if you could elaborate on how you spread the subdominant epitopes.
Yeah. The epitope spreading phenomenon really is caused by being able to stimulate the right immune response. And that immune response is Th1 CD4 T cell immunity. So for example, epitope spreading occurs in multiple sclerosis. That's why people have progressive disease with that. You have one T cell that's turned on against a particular antigen in your brain. And if you could have limited that T cell to just that one protein, you'd be able to stop multiple sclerosis. But what happened was that T cell that recognizes that protein in the brain creates that really productive inflammatory environment, and you start getting spreading of the immune response to other proteins in the brain. And that's why people with multiple sclerosis have such a relapsing course, and it's a chronic disease, and the immune response causes a lot of damage. What we're trying to do with cancer is cause that same damage, that limited autoimmune damage, just around the tumor. Now, you could do this one of a few ways. We're trying to stimulate endogenously a T cell that will be a memory cell that will stimulate a very inflammatory immune response, and that T cell is specific for proteins in the tumor. Other investigators have actually taken the cytokines that can cause inflammation and injected them directly into patients' tumors to try to get that local inflammation and that epitope spreading. But it's clear in tumor vaccines, probably about 3 or 4 years ago, one of the biggest things people were really trying to achieve with a cancer vaccine was to generate certain levels of immunity, like this is a high level of immunity, or immunity the same as what we'd see with a tetanus shot. But now we know that probably what we really should be looking at is the character of that immune response and the fact that we're creating productive inflammation with the immune response. And the most common way to measure that is epitope spreading. Any other questions? Thank you very much. Thank you very much.